Hello everybody. I'd just like to cover some summary material from the lecture on perception. The first thing we talked about in this lecture was the two-component system of bacteria. Now the two-component system is a system that's unique to bacteria that's not conserved in higher eukaryotes. Basically the system is one whereby E. coli can perceive sugars and things in the environment and is able to activate its flagella and propel itself towards the sugars. So the way this system works is the ligand, which in this case may be the sugar, binds to the receptor. In this case, the receptor is a new type of receptor that we've not come across before, and it's called a receptor His kinase. Now, the receptor His kinase autophosphorylates itself when the ligand binds to it, and it phosphorylates itself on a histidine residue. This is an autophosphorylation reaction. Then the His kinase transfers this phosphate group from the His residue onto an aspartic acid residue of another soluble protein. This soluble protein can then move to the base of the flagellum and cause a biased movement of the bacteria towards the attractant. Here's a little figure which just shows what I've been talking about. Here's an attractant that binds to the His kinase receptor, causing autophosphorylation of a histidine residue on the receptor. This phosphate group is then physically transferred onto the aspartic acid residue of a soluble protein. This phosphorylated protein can then move to the base of the flagellum and cause the flagellum to drive the bacteria towards the attractant. Okay, now we're going to look at signaling in higher eukaryotes. And signaling in higher eukaryotes uses a receptor that we've come across before, which is the G protein coupled receptor. Now previously we've looked at this receptor in context of a ligand binding, activating a heterotrimeric G protein complex, and then the alpha subunit through nucleotide exchange becomes activated, and then this alpha subunit can activate another enzyme activity in the cell membrane, such as adenyl cyclase. Now, this is how we've looked at the G protein coupled receptors before. We're going to look at these receptors now in a different context. We're going to be looking at the perception of light, so in other words, photodetection in the eye. In the human eye, we have a fairly complex system where light is perceived and transferred into electrical signals. These are then unscrambled by the brain into the perception of an image. And to do this, we need to perceive the signal and then send the signal. And then also we need to switch off the signaling from that photon of light so that we can then perceive another photon of light. So we perceive photons of light in these rod and cone cells. Now, these rod and cone cells reside in the back of the eye in a layer of cells which form the retina. And the rod cells are used in the perception of a wide range of different wavelengths of light, and the cone cells function to perceive different wavelengths of light, such as effectively colour. So here's a little schematic of an eye with the light passing through the lens and being focused on the retina. And here's a little blow-up of the retina here, showing the rod and cone cells at the back of the retina, and the light passes through a range of these synaptic cells. It is perceived by these rod and cone cells, which then act as neurons to fire a signal which effectively is sent to the brain through the optical nerve. So rod and cone cells are specialized cells with a specialized function. Now, to achieve this function, they have an outer segment and an inner segment. Now, the outer segment is a large kind of elongated bag which contains disks, and it may contain up to 1,000 of these disks. And within these disks, they're like little membranous sacs, and within these disks are the receptors that detect light. So these G protein coupled receptors are on the membranes of these disks. The inner segment of these rod and cone cells contains the normal things we would expect to see in a eukaryotic cell, such as mitochondria, nucleus, endoplasmic reticulum. These cells also contain a synaptic body which is able to transmit the electrical signal into the nervous system. So effectively, rod and cone cells are specialized sensory neurons, and light causes the hyperpolarization of these cells, which is then transferred as an electrical potential into the nervous system. Here is a schematic of one of these cells showing the outer segment, which contains these disks, and within these disks are the G protein coupled receptors. The cells also contain an inner segment which contains the mitochondria and the Golgi and the nucleus, and there's also a synaptic body at the end of the cell here which synapses into the nervous system. So the G protein coupled receptor of these cells is referred to as rhodopsin. Now, rhodopsin is a receptor that can perceive a photon of light. So effectively, rhodopsin is made up of a protein called opsin, and rhodopsin is made up of a chemical which is a derivative of vitamin A, referred to as 11-cis-retinal, and this is able to perceive light, and the photon of light causes a change in the structure of this molecule, which is then perceived by the opsin. Here's a schematic from Hancock textbook showing the G protein coupled receptor. Now, if you look here, this is not the plasma membrane, this is the membrane of one of these disks. On the cytoplasm side of the disk, we have the C-terminus, and on the intradiscal side of the membrane, we have the N-terminus. So effectively, we have a 7-pass transmembrane protein embedded in the disk membrane, and this receptor is able to perceive a photon of light. The C-terminal side of the opsin is where the opsin is able to interact with other proteins, which are the heterotrimeric G proteins. And then through transduction through the heterotrimeric G proteins, we lead to the hyperpolarization of the cell, which can then fire an electrical signal to the nervous system. Also, on the C-terminal side of the receptor, there's a region that can be phosphorylated on serine and threonine residues, and the phosphorylation of the receptor leads to the deactivation of the receptor. So here's the schematic I just showed you of the opsin molecule. Here's the C-terminus side of the receptor. Now the heterotrimeric G protein binding occurs in this region here, and on the C-terminus, this is also the site of phosphorylation to turn off the receptor. So the serine and threonine residues on the C-terminus can be phosphorylated. So photoisomerization causes a conformational change in the protein. Now this is driven by the 11-cis-retinal compound. So a photon of light hits the 11-cis-retinal compound. It causes a photoisomerization of that molecule. And effectively, this causes a conformational change in the opsin protein. So effectively, the 11-cis-retinol is a chromophore which can absorb light, and it's covalently attached to the opsin protein. And together these two make up the rhodopsin receptor. Light causes the 11-cis-retinol to be photoisomerized into all-trans retinol. And the all-trans retinol causes a conformational change in the opsin, and then this causes the receptor to be in an excited state, meaning it can then activate and transduce the signal to the heterotrimeric G protein. So this is just an image, again from another textbook. This is a nice color image showing the 7-pas transmembrane protein here, with the 11-cis-retinol embedded within the helical structure. And when the light hits this molecule here, it causes a conformational change in the opsin. So the receptor is bound to a heterotrimeric G protein complex. Now this is referred to as transducin. Now transducin is made up of alpha, beta and gamma subunits, and it's the alpha subunit which is a G protein. So in the dark, the transducin alpha is bound to GDP. And when light hits, conformational change happens in the receptor, which activates the receptor to exchange the GDP of the heterotrimeric G protein into GTP. So this just shows the heterotrimeric G protein with the alpha, beta and gamma subunits bound to the receptor. The alpha subunit is the actual G protein, which can then activate or transduce the signal to another component in the cell. So effectively, when the receptor is activated, it acts like a guanine exchange factor. So it causes the GDP to be exchanged for GTP. Now this causes activation of the G protein. The G protein dissociates from the heterotrimeric G protein complex, and the alpha subunit can then interact with another cellular constituent. In this case, the alpha subunit interacts with an inhibitory peptide of a phosphodiesterase. So the alpha subunit effectively removes the inhibitor from the phosphodiesterase, and therefore the phosphodiesterase can break down cyclic GMP. So this is a diagram explaining that from Hancock, where we have light hitting the receptor, activating the G-alpha subunit, which then leads to the activation of a phosphodiesterase, which breaks down cyclic GMP. When cyclic GMP is absent, this channel closes, and then this leads to hyperpolarization of the cell. Once these channels close, you get an imbalance in the electrochemical gradient, because there are other membrane exchanges, such as a sodium-potassium ATPase in the plasma membrane, that continue pumping ions, but these ions can't adjust their concentration now because this other pump is closed. So closure of the channel through breakdown of cyclic GMP causes a hyperpolarization of the plasma membrane, and then this hyperpolarization is then signaled through the neurons to the brain. So once rhodopsin's activated, it needs to be turned off. So it's been shown that rhodopsin can be phosphorylated by rhodopsin kinase on the C-terminal end of the receptor. Once phosphorylated, the rhodopsin can then interact with another polypeptide called arrestin, and the arrestin prevents rhodopsin from interacting with transducin, and therefore it prevents further signaling. There's a nice diagram here from Leninger, which shows some of these concepts we've been talking about. So the perception of light by rhodopsin, the activation of the transducin alpha subunit of the heterotrimeric G protein, exchange of GDP for GTP, the interaction with the inhibitor, which causes the phosphodiesterase to become active. The phosphodiesterase then breaks down cyclic GMP, therefore causing closure of this channel. There's another channel which is still pumping away, causing hyperpolarization of the cell, leading to a signal going to the brain. Now, to turn this system off, we have phosphorylation of the C-terminal residues of the receptor, the binding of some proteins leading to arrestin binding to the phosphorylated receptor, and then the dissociation of the retinol, and then the recycling of the rhodopsin. So it turns out that in addition to the perception of light, the G protein coupled receptors are also involved in the perception of smell. Now, there are a large range of molecules which we can detect as smells, and these have very different structures. And it turns out we have a large collection of genes. We have over 500 genes which code for odorant receptors. And various cells in the nose express one isoform of these 500 genes, and therefore are sensitive to a particular type of smell. These receptors are G protein coupled receptors and effectively have their influence through adenyl cyclase. It turns out that G protein coupled receptors are also involved in our perception of taste. Now, these receptors are linked to G proteins that are named gustusin. Typically, the textbook describes that 4 basic tastes can be recognized, and these tastes are perceived through different cellular responses. And we're not going to go into the details of how these movement of ions are perceived as taste. So what we have then, in this lecture series, we've covered a range of G protein coupled receptors and a range of heterotrimeric G proteins that interact to transduce the signal from these receptors. So we've looked at epinephrine with the binding of the GS subunit to activate adenyl cyclase. We've looked at the perception of light with the binding of transducin to activate a phosphodiesterase. And I've just been flagging the fact that we have odorant receptors and taste receptors which interact with various heterotrimeric G proteins to activate other enzyme activities such as adenyl cyclase or phospholipase C. So these G protein coupled receptors are very versatile and have a large family of genes that give rise to a large family of receptors used in different perception. So it turns out that hormone signaling, vision, smell and taste seem to operate through fairly similar systems. The final output of this signaling is the activation of a protein kinase or regulation of some cellular enzyme, or the change in the membrane potential, which is then passed on to the brain. And all of these systems involve a heterotrimeric G protein, and these are activated by GTP exchange and are inactivated when the GTP is hydrolyzed by the intrinsic GTPase activity of the G protein. Okay, I hope that helps with your study.